All right, good morning, happy Monday. We have some tough stuff to get into today. So get into all the nitty gritty of the adaptive immune system. So hope you brought your good thinking brain today. Let's start out with a couple of review questions from last time. So hopefully you were kind of staring at this before we got started here. Which of the following molecules encountered by our adaptive immunity? By our adaptive immunity, I mostly mean B cells and T cells, would likely be the most antigenic. Okay, so by that, I mean, which of these molecules is most likely to be noticed by our B and T cells? All right, it's kind of a tricky question. That's why I thought it would be a good one for class. Okay, so what kind of molecules did we say last time were typically the most antigenic? Like what kind of macromolecule? Proteins, right? So that would, you could cross out the capsule, the phospholipid, or the glycoprotein. That's only partially a protein. So what is about hemoglobin? Is that a protein that's is in our bodies? Yes, right? So that would be an autoantigen. So usually, as long as everything's going well, that's not something that should be noticed. We're not going to go through the mechanism of how that maturation of B cells and T cells takes place, but they will learn to ignore self-antigens, autoantigens. And as long as nothing goes wrong with that process, then we don't have autoimmune disease. Okay, so correct answer here is this viral spike protein on the influenza virus. All right. Another question that's a little bit more straightforward. We talked about the structure of the antibody. So as you're studying, maybe sometimes just okay, we talked about the structure. Can I draw it out and see if you know the parts of the antibody? The stem region, remember, that's the portion that there's 5 different variations of it. It's the FC portion. Okay, that's another name for that is stem. Okay, the FAB, that stands for fragment of antibody binding. That's the ends of the Y that actually comes in contact with the epitopes of antigen. Okay. So right, the variable region is on the FAB region, and that's made up of both heavy and light chain. So, all right, any questions from last time? You know, antigen, different kinds of antibodies, function of antibody. Okay, so where we left off last time, and by the way, I don't remember if I said this or not last time, but I'm presenting this in a totally different order than what I have in the past. So I don't know, any feedback you have on that, like, oh, this didn't make sense, and then suddenly we were doing that. I'd definitely like to hear that. But so I'm trying to follow the order that is kind of presented in your textbook. So that's the reason why I tried to change and accommodate that. So we introduced this concept of major histocompatibility glycoproteins. So we talked about that these are surface proteins that we have on every nucleated cell in the body. Remember, the only cells that I'm aware of that don't have nuclei are red blood cells. So pretty much every other cell, we've got these proteins that are going to have several different functions. There's 2 different kinds. Some of these, which we call MHC class 1, present antigen that's inside of the cell. And they're presenting that to a specific kind of T cell, a cytotoxic T cell. So we'll get to a picture of that in a minute. The other kind, MHC class 2, it's only antigen presenting cells that will have MHC class 2. So remember, that's dendritic cells, macrophages, as well as B cells. B cells aren't really phagocytic, but they have a way to kind of ingest antigen and present it again. So those are our antigen presenting cells. And they're going to present exogenous antigen, meaning antigen that's outside of the cell. And they're presenting it specifically to T helper cells. Okay, so again, typically it's not the T helper cells that are actually going to act on a cell to kill it. So it's presenting antigen to cells that play a role in activating B cells and cytotoxic T cells. All right, so it's always good to have a picture to go along with this. I'm not so much worried about the structure of these glycoproteins. This gets into a little bit more detail that I'm interested in going into, but here's our MHC class 1. Again, these are on all nucleated body cells. They have one arm here that spans the membrane of our cells. They are, again, they're presenting self-antigens. Let's write this here, presenting self-antigens as well as bits of endogenous antigen. So the idea is that this is a way for cells to advertise whether they are infected or not. So let me come back to this page in a minute. Just kind of, your book doesn't really go into very well kind of the mechanism of how that happens. This is just a very basic idea of how endogenous antigens are presented by MHC class 1. So basically, there's a complex of proteins that they call this proteasome complex that goes around all of our cells and kind of chews up bits of protein in the cell, which it seems like that would hurt our cells, but apparently not. It just kind of chews up, again, random sampling of proteins in our cell. And those epitopes, it's maybe like 15 to 20 amino acids in length, that just kind of tiny fragments of proteins are presented, find their way into the rough endoplasmic reticulum. That is where the MHC glycoproteins get expressed on the inside, the lumen of the rough ER. And those, again, sampling of epitopes that are kind of chewed up by this enzyme complex will match up perhaps with certain MHC proteins. So we have different specificity of those MHC proteins. Some fragments will match with some with others. So that's why this is kind of drawn in all different shapes. Sometimes they're going to fit, sometimes not. Okay, we get a little budding out of that rough ER into an endosome, kind of packaged by the Golgi here. And then those, that finds its way out, kind of fuses with the cell membrane. And then those MHC1 proteins are expressed on the surface of the cell for a time. After a while, they degrade and new ones are expressed. Okay, so the point is here that if self proteins are presented, then they should be ignored. Okay, we shouldn't have T cell receptors that recognize self antigen. So self proteins, self antigens are ignored. If you have some kind of virus or other endogenous pathogen, intracellular pathogen virus antigen should be noticed. Okay, so we'll talk about how those T cells get activated, maybe at the end of lecture today or next time. All right, so the mnemonic that I like, MHC class 1, if you raise your hand, if you raise one arm, you're volunteering something. Okay, so your cell is volunteering on the outside what's going on the inside. Okay, and that's, whoops, bumped something there. And that's what, again, it's how our immune system notices virally infected cells. Or there are other pathogens that are intracellular as well, other than viruses. Okay, then the MHC class 2, remember this is only our antigen presenting cells that have MHC class 2. They also have MHC class 1 because they're nucleated cells. Okay, so let's make note of that. Also will present or have MHC class 1, those macrophages, dendritic cells, and B cells. Okay, and here they are presenting antigen that they have phagocytized. Okay, so let's make a note of that. Presenting phagocytized antigen. And apparently there's a little, just to muddy the waters a little bit, there's a little bit of cross presentation. Sometimes that phagocytized antigen gets presented by MHC class 1, but we're not going to really address that because that's kind of just wades into deeper waters, but there can be a little cross presentation. Here's your books graphic about just kind of to visualize how that happens. Here's a bacterial pathogen that gets phagocytized. Here's the endosome, that phagosome, and then these little pink balls are the lysosome that fuses with the phagosome. Okay, that's where all the killing takes place. And then I don't really like the way this draws it, but this MHC class 2, these actually get expressed on the inside of that phagolysosome, because that's where all the chewed up pathogen is. Again, short amino acid fragments are recognized by that MHC class 2. And then here on the outside of the cell, that macrophage or whatever is presenting the MHC class 2. Okay, so these are the kind of cells, the dendritic cells and the macrophages especially, that are traveling around in the lymph. We'll talk more about our lymphatic system shortly. And they're trying to make connections with T helper cells. Okay, so they're presenting this antigen to T helper cells so that the T helper cells then can activate the cytotoxic T cells or the B cells. Okay, lots of checks and balances, lots of kind of immunological synapses that have to take place. We want to make sure that an adaptive response is appropriate. We don't want to start ramping up, antibodies or killing our own cells when that's not appropriate. So it seems like, again, a lot of checks and balances. So if we have, MHC1 raising your hand, volunteering, MHC class 2, maybe cells, with 2 hands here. This is what I'm dealing with. I need some help with this. It's not as perfect and mnemonic, but they're going to present to other kinds of cells. This is what I just chewed up. I need some more help with this. Okay, that kind of helps me because you do need to keep track of, which is which, and that would be helpful later on. Okay, so let's stop for another review question before we move on here. Again, this is a question that I've asked on an exam, but it's a little bit tricky, so I'll share it with you now. Take a minute. a little on the wordy side, but I like this question because hey, we talked about Plasmodium, we talked about malaria in the past. Hopefully this kind of looks familiar because I just reminded you of that, but it's kind of an interesting thought. What about pathogens that infect red blood cells? Okay, they do not have MHC class 1, and therefore our our B cells and our T cells don't have a way to recognize if there is an infection going on within the cell. Okay, our antibodies, they're not going to do any good for a pathogen that's inside of the cell. So it's problematic. Our immune system has a hard time fighting that off. And sometimes some infections have ways to downregulate the cell's production of MHC class 1. And so that's a problem as well. Pathogens are always it's always like an arms race, right? They do one thing and we do another. All right, so we're gonna continue kind of putting the pieces together here, and we're kind of leading to how B cells and T cells get activated. And we've talked a little bit about what they do when they're activated, but we'll talk about more. Okay. Let's just see here. Okay, so just pause for a minute. We talked about kind of hematopoiesis of different kinds of formed elements in the blood. As we zero in on B lymphocytes here, let's talk about where they come from. B lymphocytes and T lymphocytes as well, actually. So we'll write that here. B and T cells are going to be formed in the red bone marrow. That's where we have lots of stem cells. Okay, and B cells are also going to mature in the bone marrow as well. And I should say that's where most of the B cells are made. So let's leave it at that. Not all of our bones have red bone marrow in it. It's mostly kind of our bigger bones, some of our flatter ones like the scapula, the head of the femur here. That's what this is showing, I believe, that kind of ball that fits into our hip bones there. Okay, so that's where we find some red bone marrow. So that's where the B cells originate, where they mature. What do I mean by mature? That's when the B cells will get their B cell receptors. And we're not going to go into the mechanism of how this happens, but there's a process by which B cells that have B cell receptors that recognize self-antigen are just slated to be killed or to die off. Apoptosis, self-suicide. So we end up with hopefully a repertoire of B cells with B cell receptors that don't recognize self-antigen. And if you look at the structure of the B cell receptor, it should look familiar because that's exactly what an antibody looks like, with the 2 heavy chains, 2 light chains. The antigen binding site here is on the outside, and the very ends. Okay, so like with the antibody, both variable regions are going to recognize the same antigen. Or epitopes of antigen, same epitopes. Okay, so it seems like it would make sense to have B cell receptors on one B cell that all recognize something different. And then that B cell could maybe find the antigen that it's specific for a lot easier. But that's not the way it works. On a B cell, there's something like 100,000 B cell receptors per cell. So they're all over the outside, and they all recognize the same thing. So the same antigen. So it's kind of the analogy that I see, B cells are kind of like snowflakes. Like they all have different B cell receptors, which is almost true. We think that we only have maybe a few B cells in our body that would recognize a certain antigen. So if that B cell, those B cell receptors do recognize the antigen, then our cell has ways, our body has ways to make many more of that B cell with that specificity. Okay, so it's induced. We call that a clonal expansion. Okay, and this does make sense when you understand that if a B cell receptor does recognize an antigen, that sends a very weak signal. Just there's one arm that recognizes it, and actually lots of B cell receptors on the surface of the B cell all have to recognize. Not all of them, but many of them recognize the same thing, and that sends a very strong signal then to kind of begin the activation process, I guess you could say. Okay. All right. So a question that plagued researchers for a really long time is, if we have all these different B cell receptors that recognize all these different things, how is that possible? Because we only have about 25,000 genes in our body. So how can we make millions of different B cells? Well, the basic idea is, and I know this is kind of a complicated looking diagram, but the basic idea on the gene that encodes for the B cell receptor for that antibody binding region of both the heavy chain and the light chain of the B cell receptor, the gene has three different kind of variable regions. There's a V region, a D region, and a J region. And that V region has 65 different possibilities that could actually get translated into the B cell receptor. The D region has 27. Don't memorize those numbers. The J region has 6 different possibilities. So, and basically, there's this RAG protein or an enzyme that goes down and picks out one of the V, one of the D, and one of the J regions. Okay, so we call this a VDJ recombination. Okay, so it's like somebody going to get dressed in the morning and they have 65 different shirts and they randomly pick one, 27 different pairs of pants, they randomly pick one of 6 pairs of shoes, and then they get dressed. Now, sometimes that outfit is not going to work. Your roommate might say, you can't go out dressed like that, go change. Well, sometimes the combination that RAG protein picks is not a viable protein. Okay, so that B cell just kind of dies by a programmed cell suicide. But assuming that it's a combination that works, okay, that is what is incorporated on both of the antigen binding regions of that B cell receptor. So with this recombination event, both happening in the heavy chain and the light chain, we think if you do the math, there's over 9 million possible B cell receptors with VDJ recombination alone. And then we think there's other kind of random recombinations and mutations that put this number up over 10 to the 23rd power. So millions and millions of different B cell receptors. And a similar process happens with T cell receptors as well. Okay, so the main idea is that we don't have one gene for every B cell receptor. It's these recombination events that can kind of put this great, randomness to our B cell receptors. Okay, so that brings us to how are B cells activated. And remember what happens when they're activated. Let's talk about that first. I'll skip down here. B cells, they get a little bit bigger. They start to rapidly divide, and they're going to differentiate into plasma cells. Okay, so it's the plasma cells then that secrete antibodies. And then sometimes we also get memory cells. It depends, though, how those B cells get activated. Okay, so there's 2 different ways the B cells get activated, they can be activated by antigens that do not require T helper cell help. Okay, so we call those T independent antigens. Okay, so when the B cell recognizes the antigen, that's the primary thing that it needs to happen, and it will start to kind of differentiate into plasma cells. The other way that we think is much more common is that it's activated by antigens that do require help from T helper cells. So that's called T cell dependent antigens. Okay, so I know the wording is a little bit confusing there. Hopefully you get used to it. Let's talk about that first way because that's a little bit easier. Okay, so antigens that will stimulate the B cells to activate directly. That doesn't need to be any additional signals from T helper cells. Well, before I get to that, here's a plasma cell of the guinea pig, and I think this is just the coolest picture because, you know, normally when you see a eukaryotic cell, you draw one of everything. When that's not actually, it depends what kind of cell it is. With a plasma cell, you can see there's a big nucleus in the center of the cell. And I think these little bluish blobs here, I think those are mitochondria. There's maybe a few Golgi bodies in there. But most of the cell here is taken up by rough endoplasmic reticulum. And that makes sense because remember that's studded with ribosomes. So cells with a lot of rough ER, they are making proteins that are destined to be secreted outside of the cell. Okay, so lots of rough ER. Plasma cells secrete about 2,000 antibodies per second. So they are really kicking out those proteins, those immunoglobulins. There actually are different kinds of plasma cells. These ones that are really kicking out antibody fast, they're pretty short-lived. They only last maybe a couple days. There are longer acting plasma cells that make antibody more slowly over a longer period of time. Okay, so again, that's a transmission electron microscope of a plasma cell. All right, so the first way that B cells can get activated to differentiate into plasma cells is that they don't require help from T helper cells. So T cell independent antigens. What kind of antigens are these? Typically, they are large non-protein antigens with lots of repeats, oftentimes a polysaccharide. And remember, we said before that usually polysaccharides aren't very antigenic. They don't very often stimulate a very good immune response. There are exceptions. Okay, we often find polysaccharides an example of a place we find them is in bacterial capsules. Okay, if you remember back, if you're in lab, when we stain bacterial capsules, I know I talked about with my class, we like to stain the background because it's hard to find dye that will stick to these polysaccharide capsules. They don't have a charge. So we stain the background and maybe the cell wall so that the capsule stays unstained. Okay, so that's one example. One advantage of this method of B cell activation is that it's really fast. You don't have to try to find a T helper cell that has that same specificity. So that's a process that takes a little bit longer to make all those immunological synapses. So basically, again, the B cell receptors recognizing the capsule, and it starts to differentiate into plasma cells right away. There are a few problems with this that make it not as strong a response. We don't get any no class switching. Okay, so what's the first antibody that's made typically in response to a infection? Talked about that last time. Anybody remember? I hear some mumbling. Typically IgM are the first ones that's made. Okay, so we get a really fast response here, IgM, but one problem is it's not a strong enough response to get the cytokines that will cause class switching to take place. So we never get change into IgM or IgA or IgE, ones that are more maybe suited for that particular infection. Okay, so it's only IgM antibodies that are produced, and we get no memory cell production. And maybe that's okay because it is a faster response. But the next time that, say, bacterial cell, that bacterial capsule, it's going to take the same length of time. Another big drawback in terms of if we're trying to defeat pathogens is that this type of response, it doesn't really take place very well in young children. How young? I was trying to look at, I've heard two different numbers, below the age of 2 and below the age of 4. So, you know, younger than, we'll say, about age 4. These kids don't have much of a T cell independent response because their B cells are immature. Okay, as adults, we have more mature B cells apparently, and we have a really good response here. So pathogens that have these T cell independent antigens, it's really important that we do what we can to protect young children from these pathogens. So one example of a pathogen with a T independent capsule around it is this bacteria, Haemophilus influenzae. That's a pathogen you don't hear that much about because, again, adults don't have any problem with it. We named it Haemophilus influenzae because 100 years ago, with that worldwide flu pandemic, we thought this bacteria might be what was causing that. Astonishing to think that we had such a pandemic and we didn't even know what pathogen it was that was causing it. Of course, we found out later it was a virus, but at the time we thought that it might be bacterial. This pathogen doesn't cause the flu. It causes a bacterial meningitis. And there are several different kinds of bacterial meningitis, but it's a very serious infection for young children to get and can cause, you know, lifelong disability if they survive it. So really important to get young children the vaccine to that. It's called the HIB vaccine, Haemophilus influenza B. Question?
So are you saying that this targets the T independent B cell?
So good question. What I'm saying is this infection has polysaccharides that act as T independent antigens. So our T helper cells don't help with this kind of thing. It's the B cells that have to recognize it. So they don't recognize it very well. That's the point. Okay, so you know, sometimes, you know, adults think, why do I need to immunize my baby for this? I've never heard of this infection. I've never had it. Yeah, as adults, we don't get it because, again, assuming our immune system is working well. So that's one of the first, I think at like 2 weeks of age, it's one of the first ones that babies get, that Haemophilus influenza B. And it's like 4 different shots at least. Okay, so this is a little bit less common, these type of antigens, but they do have a little bit different response. So here's just a graphic. Here's a B cell that's, again, recognizing some usually a polysaccharide antigen, maybe on the outside of a bacterial cell here. And that causes it to differentiate, the B cell to differentiate into plasma cells and to start kicking out IgM antibodies. Okay, no memory cells. Again, no memory cells drawn there. Okay. So make a note of that. Remember, no memory. It's supposed to be a Y there. Okay. All right. So let's take a look at the other way that B cells get activated. And this is, again, the most common because this is the way all protein antigens are going to activate B cells, and that's called T cell dependent antigens. Okay, so I tried to kind of write down some of the steps here before we get to the next graphic to kind of walk you through it. The T cells, the T helper cells also technically have to be activated to recognize the same antigen. And then when they are, they're going to differentiate into these T helper 2 cells, and they will secrete cytokines that will activate the B cell to differentiate into plasma cells. Okay, and then we get really a fuller response because we get better antibody eventually produced. Okay, so let's walk through this. Your book simplifies this a little bit, which is fine. But so here we've got a B cell that's recognizing some epitopes on some bacterial cell, for example. At the same time, there's going to be some helper T cells that at some point get activated. This graphic kind of leaves out that step. But when a B cell encounters this antigen, it actually has a way to kind of take in some of that antigen and present some of it by MHC class 2. And we won't go into the mechanism of how that happens, just that B cells also present with MHC class 2. And remember, it's helper T cells that recognize the antigen when it's presented by MHC class 2. So, and helper T cells, we haven't talked about this yet, also have this CD4 glycoprotein. That CD4 actually, this is going to bind to the MHC class 2. So it kind of stabilizes this immunological synapse between the C cell receptor and the MHC class 2 that are both recognizing that same epitope of antigen. The CD4, again, stabilizes that. This helper T cell, which has already been differentiated into a T helper class 2. I've got a graphic later that actually your book shows how that happens later. When this synapse is made, it will start to release cytokines. So that's what all these little dots are. And we won't worry about which cytokines they are, but they act on the B cell to proliferate. Okay, we call that clonal expansion. Okay, because again, they've gotten the right signals from the T helper cells. They're going to make more of themselves. That's happening over here. Lots more B cells that differentiate into plasma cells that make antibody. And we also get some memory B cells with that same specificity. So the idea is with those memory cells, the next time they recognize whatever, this antigen, this epitope, then they don't need those signals from the T helper cells anymore. So they get a much faster response. I think so the next graphic shows that. So Again, this is T cell dependent primary versus secondary response, adding a little bit more. We talked about this before, we're adding a little bit more detail to it. So the first time we're exposed to that antigen, it takes quite a while till antigen gets made, because again, you have to find the right T helper cell with the right T cell receptor to that's recognizing that presented antigen. So it takes at least 10 days to start making antibody. IgM is that first antibody with some class switching, then that will change to most often IgG, but it might be some other kind of antibody that's better suited. And then at some later date, if the host encounters the same antigen, you get that IgM production from those memory cells, according to this timeline, about in half the amount of time. Okay, so maybe 5 days, but even more importantly, you get class switching sooner and a much larger amount of antibody the second time. Okay, so again, that's due to the memory cells. Okay, they don't have to go through all the kind of laborious activation that they had to before. They're primed and they're ready to go. Okay, so why does that take so long to get antibodies made? Well, that leads us to a little bit more thorough discussion of our lymphatic system, and then we'll get to how T cells become activated and what they do. So this might be a little bit more familiar. Probably a lot of you know your anatomy a lot better than I do. But the lymphatic system involves a whole network of lymph vessels this is going to be kind of the highway by which our white blood cells are going to flow through and make these immunological synapses with antigen presenting cells and T cells. So they're traveling through the lymph. We talked about this already. We'll talk about again the primary lymphoid organs. That's the red bone marrow. Again, that's where B and T cells are made. And the B cells mature. The other primary lymphoid organ is called the thymus. And this is where T cells mature. Okay, so the thymus is this little gland that kind of sits on top of the heart. Most of that T cell maturation takes place before a baby is even born. Okay, so that thymus tends to shrink apparently as a person ages. Okay, so those are the primary lymphoid organs, red bone marrow and the thymus. Lots of different secondary lymphoid organs, and that's where lymphocytes are activated. First thing we think about are the lymph nodes. We have somewhere on the order of 500 lymph nodes throughout our body, and they are mostly about the size of a pea. Okay, so they're pretty tiny most of the time, although they can enlarge a little bit. And this is the lymph nodes is primarily again where a lot of these immunological synapses are going to take place. Also, the spleen. Its primary function is to filter blood, where lymph nodes are going to filter lymph. We talked about one of the functions of antibody is to clump antigen together. Okay, so that antigen can get filtered out in the spleen. And again, when it's big clumps, we can survive without a spleen. Okay, so if either by, maybe a person has some kind of blood infection and it has to be removed, or sometimes an accident will cause a rupture of a spleen. Apparently you can't just sew a spleen up and have it be good to go. It usually has to be removed. So people without spleens are they're a little bit more susceptible to certain kinds of infections, especially bacterial infections. So people without spleens might take antibiotics prophylactically. So just if they know they've been exposed to an infection, they might need more vaccines that more often than what other people would. But they typically can get along fine. Of course, people get along fine without their tonsils as well. Sometimes if people have particularly large tonsils or really deeply creviced tonsils, then bacteria can kind of get caught in there, and people can have, especially get strep throat a little bit more often. We've got lots of, basically the tonsils are basically a lymph node. We can do without them just fine. Got lots more. And then the malt. This is another kind of secondary lymphoid tissue that basically lacks the hard outer covering of a lymph node. It stands for mucosal associated lymphoid tissue. So we have lymphoid tissue on all of our mucous membranes. The one that I'll highlight here, it's called Peyer's patches. We have this mucosal associated lymphoid tissue in our intestine, whose job it is to kind of randomly sample what's passing through the intestine. So the Peyer's patches are really important to kind of notice, we'll say, foodborne illness. Okay, so randomly taking samples there and seeing if there's any antigen that matches up with the B and T cells there. Okay, so here's, I don't know why I included this. This was just your book's picture of some of the, well, the thymus, how it sits kind of on the heart there. I think what's drawn here, and maybe correct me if I'm wrong, some of you that are a little bit more up on your anatomy, I think those those blue ducts there, I think that's supposed to be the subclavian vein. Oh, good. Maybe that's it. Okay, because it doesn't tell what it is there. But as we'll see, when the lymph, when it goes in one direction throughout our whole body, and it dumps into the part of our body where the blood pressure is the lowest. Okay, so that's the subclavian vein. Okay, so again, lymph basically dumps here back into our bloodstream. What is lymph? I haven't addressed that yet. Here's a close-up of a blood capillary and how it's in close association with the lymphatic capillary. We talked about how, especially during inflammation, those blood vessel walls will get a little bit leaky. So we get cells, again, white blood cells, leaving the veins by that process called extravasation. That's happening even more during inflammation, but normally we've got, fluid leaving the blood vessel walls, gases. Okay, when the pressure in the tissues in between there is greater than the lymphatic vessel, all those fluids and cells and things will enter into the lymphoid capillary. And like I said, we don't have a big lymph pump. That lymph will move in one direction, and it moves by smooth muscle contraction. Okay, let's see. Moves. And we have valves here in the lymph, kind of here and here, that make sure that it only moves in one direction. And again, it moves towards the subclavian vein, which is right up there, right below our collarbone, and it dumps back into the bloodstream there. Okay. On its way, it's going to encounter lymph nodes every now and then. Okay, so this is we've got several different afferent lymphoid vessels. So afferent means towards. You see there's several of them, 1, 2, 3, 4, and only one efferent lymphatic vessel, E meaning away from. Okay, so it's kind of a bottleneck of lymph there. It gives that the cells that are in lymph a chance to mix with other cells. All right, we'll pick up there last time. We'll probably start vaccines. Sorry, we'll pick up next time. We'll start vaccines on Wednesday and finish up T-cell activation.